The overall goal of this protocol is to biochemically characterize tRNA-Isopentenyl Transferase activity in vitro.
This method is useful for the in vitro detection and characterization of tRNA-Isopentenyl Transferase activity of purified recombinant enzymes. The main advantage of this technique is that it is a simple and direct assay used to detect a covalent RNA modification.
Though this method provides insight into tRNA-Isopentenyl Transferase activity, it's potentially adaptable for the biochemical characterization of a wide range of RNA-modifying enzymes.
The first step in this procedure is to thoroughly clean the glass plates that will be used for casting the gel. Wash the plates with soap and water, rinse well with deionized water, and then clean the plates with isopropanol and lint-free wipes. Next, assemble the plates and spacers. Mix the following reagents to make a 100 ml 20% acrylamide, 7.5 molar urea, 1x-TBE gel, 80 ml of urea gel concentrate, 10 ml of urea gel diluent, and 10 ml of urea gel buffer. Add 40 microliters of TMed and 800 microliters of freshly prepared 10% ammonium persulfate to the mixture. Draw the gel solution up into a large syringe and dispense the gel solution between the glass plates, tapping on the glass while dispensing the solution to prevent bubble formation. Allow the gel to solidify at room temperature for 30 minutes. After the gel is solidified, clamp the gel onto a vertical gel apparatus.